Now we have the pleasure of welcoming Dr. Triparna Sen with the Icahn School of Medicine at Mount Sinai. Let's give her a round welcome.
Good morning, everyone. Let's do better than that. Good morning, everyone. There you go. I am Dr. Triparna Sen. I'm a translational lung cancer researcher. It's a mouthful, so I'm going to tell you what that really means. What does translational lung cancer research means? So I run a lab which really studies the biology of lung cancer, primarily small cell lung cancer. I've been doing this for over a decade now. And have any of you read the book called Ikigai? Raise your hands if you have. It's a book that talks about your purpose, your life's purpose. You are the happiest in your life when your interest, your expertise has met your purpose. And I'm extremely privileged to be standing here today because I'm going to talk to you about my purpose. My purpose is lung cancer research. I live and breathe lung cancer research. And my family knows that you cannot take me on a vacation. And that's not only because I suck at work-life balance. I do. But it's because what I've left behind in the lab is so important to me. It's important to me more than probably a lot of things in my life. And I'm telling you this, I'm really humbled to be here, and I'm going to tell you what we do. Jane and Kimberly, you asked two questions when Trudy was speaking, and I'm going to try to answer those questions for you here. So what we do, we study lung cancer. We use a lot of cool techniques. We use cells, we use mouse models, we use techniques to look at how drugs affect cells, how cells affect tumor, how immune cells affect tumor, tumor affects immune cells, all of that. But with the goal of precision medicine, that's always the goal. Novel targets, novel biomarkers, that's always the goal. And I wear another hat. I'm the director of the Lung Cancer PDX program at Mount Sinai, which means I collect tissue and blood from patients who walk into the clinic, and I try to make mouse models and other organoid cell lines. You might have heard these words from Trudy's wonderful, elegant talk. And I try to study the disease, the biology, so that we can come up with novel therapies. That's basically what I do. And my goal is to take the knowledge from the lab to the clinic, and then learn from what we have learned from clinical trials to then come back to the lab and then make it better. That's an ongoing work. And I'm going to talk to you about what we do, because it can get a little, jargony, because, and why does it matter? What have we learned from the tissues, from the blood that we have collected from you? How has it changed the field? How do we have all these trials that you're hearing are positive trials? Everything we have learned from you. So I'm going to talk a little bit more. So if you can think about a translational roadmap, so to speak, of small cell lung cancer. So all the way from treatment to sample collection, to preclinical research, to clinical trials, and then we go back again. What does this roadmap really look like? What it means, and how it has really progressed the field. So let's talk about diagnosis and treatment. I'm going to leave the diagnosis for my clinical colleagues because they'll do a better job. I'm going to talk to you a little bit about the current standard of care for small cell, whatever is approved right now. So most patients who come into the clinic, and I'm so glad to hear so many limited stage small cells in this room, and my heart is really full right now, but most patients who come into the clinic actually come with extensive stage disease. They come with aggressive disease that has already metastasized to various organs in their body. And the standard of care for that is chemotherapy in combination with immunotherapy. That's what they get. The initial response, the objective response rate is more than 60%, which means patients do well. they do respond to the initial line of treatment. They stay quite well for a few months. But as I heard from some of you in the room, the cancer unfortunately comes back. And when it does, as you heard from Badri, that lurbinectedin really changed some of that in the second line. But there is slim pickings when it comes to second-line treatment for small cell lung cancer. There's not really a lot of things out there that are currently approved. The patients do great. So this is the platinum doublet and the two antibodies that are actually approved in the immunotherapy setting, atezolizumab and durvalumab. I'm not going to go into the trials. You can read about them. And then when the patient relapses, this is the treatment options for them. It depends on how they have responded to the chemotherapy. It depends on how many of months, and depending upon that, you can either go on a clinical trial, you can get the standard of care, or you can get retreated with chemotherapy. These are all great options, and your physician should decide what's best for you. So one of the milestones in small cell has been really when immunotherapy got introduced, right? Small cell was being called a graveyard of clinical trials. This is where clinical trials come to die, right? That's used to be the thing for a very long time. But that all kind of changed to a little better in 2018 when immunotherapy got introduced. And I'll tell you in a bit why that matters. So as you see here, two trials, Empower 133 and Caspian. These names may have come up a lot of times. These were positive trials. And after a very long time, small cell saw positive trials. And that led to the approval of immunotherapy in frontline for small cell lung cancer. It was a great day for us. So where were we in 2009? All patients were getting chemotherapy. And this is sort of the survival curve that you can see here. And in 2009, the 3-year survival for small cell was less than 5%. And essentially, there were no long-term survivors. A minority, a real minority, that we called long-term survivors with small cell. Cut to 2022, this number has increased from 5.8% to 17.6% 3-year survival. And the curves do flatten out. It is a minority. Thank you very much. But don't clap yet because I want to do better. So just say you're not doing good enough. I want to know. I want to hear that. That's not good enough because it really is not. The curves do flatten out. There are long-term survivors now, both from Empower 133 5-year readout and Caspian 5-year readout. There is a minority of patients who actually do better. And they have long-term survival. So if you can take home anything, I know a lot of people are taking notes, and that's amazing. Immunotherapy does work for a minority of patients. They work, and they do have a role. It's for us to figure out who are these patient population that are having this extreme response from immunotherapy, and why are they doing better? And for the patients who are not, what can we do for them? That's the whole question. So now, you both asked, Kimberly, you asked, and Jane, you asked, what do you do with our tissues? Wonderful question. You should know what we do with your tissues. And I'm going to go over what all we do with your tissues in the lab. So this is what we call sample collection and preclinical model generation. So when you go to an academic center, if you're being treated there, you'll get a consent form. They'll come to you with a consent form. They'll say, we'll get your tissue, we'll get your blood, and it will be used for research purposes. So I'm going to tell you what that means. There is some scientist out there in the lab who's going to do a few things with your sample. So what I've built at Mount Sinai Central Lung Cancer PDX program, I'm definitely not the first one to do this. Charles Trudon and Memorial Sloan Kettering, Lauren Byers, and MD Anderson, Caroline Dive in the UK, they have done it better, bigger, and earlier than me. It's just, it's something that we do and something that is useful. And I'll tell you in a moment why that is useful. So we support the thoracic oncology services and help the entire team to generate centralized, curated library of lung cancer patient-derived xenograft models that is accessible to investigators and to the scientific community. The samples that we collect, blood, surgical biopsies, pleural fusion, biopsy samples, autopsy sample, EBUS, and also MET samples. So these samples can be directly taken for research, and I'll tell you what we do with that, or it can be injected into these immunocompromised mouse models, because we want to take mouse models, and I'll tell you in a bit what we do with those mouse models. And they grow tumors. These mouse actually become our surrogate, where we can study drugs in these mouse models. And both the tumors from the mouse and from our primary tumor from the patients can then undergo any variations or all of these platforms, right? And I'll go with each of these, what they mean, what have they told us. Histology, whole exome sequencing, proteomics, RNA-seq, single cell. Any combination, and we do all of these actually. So this is the next phase. So we have built these models, we have collected your tissue. What we do next is preclinical research. It's the research that is done in the lab. Histology, very important, and I'll tell you in a minute why. So we published this large sample set of 174 small cell lung cancer cases that were analyzed with just histology. What histology does is puts your tissue under a microscope, and we can look at it, and we can look at the morphology of this tumor. This tells us, that helps us in diagnosis. So it tells us this is small cell, not small cell. And it also tells us what are the other markers that your tissue is expressing. And based on those markers, we hope it could be a marker, and I'll talk about targets. It could be something we could target, or it could be something we could select patients with. So that helps. And what we learned from this study is very important. So these were 3 tumors that expressed all these markers. You don't have to remember any of that. But if we take the same tumor, you can see that both these markers are being expressed. So what are the lessons that we have learned from histology in small cell? Small cell is like small blue cells under a microscope. That's how the diagnosis is done. It expresses neuroendocrine markers. Trudy spoke about that. I'm not going to talk about that at all. And it expresses specific markers that I'll later talk about when I talk about biomarkers. And small cell is heterogeneous. I think that was really striking to us, that this is not a tumor that expresses just one marker. It expresses multiple markers. And small cell is more heterogeneous, which means one-size-fits-all approach may not be effective for all patients. That's what we learned. What did we learn from mutation? We learned that's not the way to treat small cell. Because small cell comes with a lot of mutations, a lot of them. And these are all loss of genes. And loss of genes cannot be targeted, right? So the path that non-small cell field has taken is targeting mutations. That doesn't work for small cell. There are no mutations to target. So what genomic analysis told us is that it has a high tumor mutation burden, major genomic alterations, loss of function mutations, but the most important message was, this is not the way to target small cell. There are no genomic drivers that are present in enough population of patients that we can target. So this is your take home from the mutation analysis that we did from the tissue that we got from you. What did proteomics tell us? So this was a study that was done by my previous mentor at MD Anderson, Lauren Byers. So she and John Haymack, they looked at small cell lung cancer cell lines, asked a very simple question. Proteomically, how do they look different from non-small cell, right? Very simple question. Very profound answer. Small cell lung cancer is different from non-small cell lung cancer at the proteomic level. They're different. And they do express proteins that are the downstream of a transcription factor, not important, E2F1. What they did was introduce all these proteins, which I'll talk about later when I talk about targets. These are all targetable. So what that proteomic analysis told us is that the protein profile is different from non-small cell lung cancer. High expression of proteins that are involved in DNA damage response. And the good news was these are all targetable. We can have drugs that we can target them. And identification of major targets, these are all in clinical trials. PARP, ATR, V1, CHEK1, Aurora, PLK1, all targets for small cell. So that's what proteomic analysis told us. Again, your tissue was the starting point for a lot of these analyses for comparisons and confirmation. RNA sequencing is how we look at the transcriptome. What did that tell us? Oh, it told us a lot. Shruti showed you this plot. This was a field coming together to kind of put together what RNA sequencing has told you. This is bulk analysis. And they also showed that some of these subtypes expresses MYC, and Shruti has been really instrumental in establishing the importance of MYC in small cell lung cancer. Very important biomarker. And DLL3. And why is it important? Because I'll talk about tarlatamab in a minute. And I know, Kimberly, you would be very interested to know about the studies that are coming out. So I'm going to talk about that. So what did RNA-seq tell us? A lot of things, actually. The expression of small cell subtype markers, ACL1, NeuroD1, PAU2F3, YAP, they're important. And I'll tell you in a minute what we are doing with this knowledge. Expression of specific markers that dictate the biology, immune genes, Notch, MYC, DLL3, they're not just names, they actually dictate the biology of your tumor. They tell you what we can target. And the expression of markers that are potential drug targets, again, PARP, EZH2, LSD1, and DNA damage response genes, they are there, and the tissues that we collected from you gave us the knowledge that these are the markers, these are the targets. Now we've got really ambitious. We wanted to see at the single cell level. Tissue was no longer enough for us. As technology advances, we wanted to look at the information at the single cell level. Why? So if you think as your cells as this like cup of fruits, all has a very, very different flavor, right? When you do bulk analysis, it's essentially a smoothie. Tastes great, but you don't know what is in there all the time. And if something is in there at a very low percentage, you will miss it. It may be important, but you're going to miss it because it's a smoothie now. So we wanted to look at it individually. That's why we invest in single cell sequencing, which is very expensive, by the way. So single cell RNA sequencing, we published the first atlas for small cell lung cancer, single cell RNA sequencing. What that told us, please don't pay attention to any of this because it's just a lot of pictures. What that really tells us is that the small cell lung cancer cells, they're very promiscuous. They don't like to commit to a state. They have commitment issues. They shift from one subtype to another, one biology to another, and they shift depending upon the stage, whether they're metastasizing, what the drug pressure is. They just don't like to stay still. That's what we learned. We learned about a minor population or a rare population that could define the biology, which we would have missed in a bulk analysis. And as more and more of these single-cell analyses are done on patient tissues, these rare populations which we are missing in bulk analysis will come up that may dictate your biology. And the most important is the interaction of the immune system with the tumor cells. I think that is a knowledge that we miss when we do the bulk analysis. But single cell tells you. Single cell tells you what your immune cells are doing in that whole milieu. What is their contribution? How are they reacting throughout this population? So that's what we learned. What did we learn? Heterogeneity, again, we knew that. Novel small cell subtypes, cell type, rare cell populations, and then a dysfunctional T cell and myeloid population in small cell. Very, very, very important knowledge, because then we know what is wrong with these tumors, how we can make them better. And then finally, what do we do with this mouse model? This was a study that came out of MD Anderson. It was led by Alison Stewart. And what they showed is that why do we put these into tumors? So first, let me tell you that no mouse model is the perfect mouse model. Everything comes with its limitations. But the most interesting thing that came out from this study is that this patient from which the sample was collected, they had a leptomeningeal disease. So the disease progressed to the brain. When the tumors were implanted into the animal, in the animal, it went to the brain. So it was actually recapitulating the patient tumor. And it was also recapitulating the treatment response. So this was collected from a patient that was actually responding to chemotherapy at that point, and the tumor actually responded to chemotherapy. So these are very, very faithful models to then do these preliminary trials in the lab to look at effective drug targets, to look at effective biomarkers. So the biology of these mouse models, they really recapitulate the biology of patients. And that gives us confidence in the predictions that we made. Is it fully translatable? Not. We're missing the immune population, right? So often what we cure in the mouse doesn't work out the same way in patients because there are limitations. But the biology does recapitulate. And it also recapitulates the treatment response. So, we have published many papers where we have done this in the clinic, in the lab, and then we have taken it to the clinic. So all of this that we do till now is for the only purpose is to take it to clinical trials. We want it to benefit patients. That's the only goal. That's what we do. And there are two pillars of designing an effective clinical trial. Your target. Your target needs to be effective, durable, and it needs to give you the quality of life that you need, right? Because it's not just about survival, it's about life. So it needs to be what fits you, right? And then biomarkers. How do we select these effective patient population that then they would respond to the therapy? So these are the two pillars of an effective clinical trial. How are we doing with the targets in small cell? So immunotherapy curves, they're not as effective as we would want them to be. So the question that we ask every day is how do we raise the tail of the curve? That's the question, right? But I just want to go back to Jane and Kimberly. Did I answer your question about what we do with your tissue and how your tissue helps us? So thank you. Yeah.
Inside.
Yeah. But thank you, ma'am. I'm going to talk about tarlatamab in a minute, but yeah. And I know Simon is going to talk about liquid biopsy. We are slowly but surely progressing there, so stay tuned. So the question with targets is, how do we make immunotherapy better? Because if you can reorient your body's own immune system to fight your cancer, that is an effective and a durable way to actually treat this disease, right? So immunotherapy for small cell, there are pros and there are cons, like with everything in life. The pros are the high mutation load and the genomic instability. But the cons are they have very low infiltrating T lymphocytes, and they have low MHC class I expression and low PD-L1 expression. So that makes them, it's actually a paradox that how does a cancer with such a high mutation burden have such low infiltration of T lymphocytes and MHC class I? It's a conundrum for us. For you, it's just bad news. But it is the conundrum that we are fighting with. So how do we overcome that? So the question is, small cell lung cancer has very low expression of MHC class I. It's what makes your tumor visible to the immune system, because your immune system needs to know there's something wrong happening to go and attack it, right? So there is something in small cell that hides it from the immune system. So the question is, how do we make your tumors visible to the immune system? So for that, we published some research 2 years ago now. That's old. And what we proposed is that epigenetic modifiers. So you've heard about genes, and then there are master controller of genes, things that control your genes. Those are the epigenetic modifiers. So epigenetic modifiers can be targeted, like LSD1, EZH2. You will hear these names all the time. There's nothing in a name. But we can target these epigenetic modifiers to then re-express MHC class 1. What that led to is this clinical trial. There are 3 trials now with LSD1 inhibitor in small cell lung cancer. So this is truly a bench to bedside, and hopefully we will learn from this clinical trial. So we'll learn a lot about whether LSD1 and EZH2 they're working or not. Another strategy, so as I said, there are low lymphocytes, so there are CD8s, and CD8s are good T cells, and they're not getting into your tumor. So what can we do to make the cold tumor, so anything that doesn't have a lot of CD8s, we call it the cold tumor, and then when CD8 infiltrates, it's a hot tumor. So how can we make this better? So for that, what my lab has been working on for many years, and now my lab has been working on for many years, is targeting this group of proteins called the DNA damage response. And I don't want to get all sciencey, but I do want to tell you that small cell, with all its genomic instability, presents a very unique opportunity for us to target DNA damage response. It's almost like it's an Achilles heel. It's that one spot that we could hit. It's one of the many spots, but it's one very effective spot that we could hit that would make these tumors shrink. And that's targeting these proteins that control the G2M checkpoint in a cell cycle, or the PARP inhibitions, which controls the repair of DNA. And what we then went on to show is that when these tumors are targeted with DNA damage response, it actually helps recruit T cells. It makes the cold tumor hot. So then now when you combine it with immunotherapy, you can really make an effective strategy for that. And there are many trials. So this was the first study that I published when I was a postdoc, and with CHEK1 inhibitor, and then we went on to talk about WEE1 inhibitor. Lauren had published on PARP. This was the first phase 1 with a PARP inhibitor, talazoparib. And since then, there have been many. There are pharma partners in the room, and I know many of them have these compounds that are currently in their clinical pipeline. They are in many phases of the trial with DDR inhibitors. We'll have to see how they do in the combination setting. And these are the trials. There are many more, actually. This is a redundant list at this point, that are looking into DNA damage response in combination with immunotherapy in small cell lung cancer. Right? So, we think it's an effective strategy. There are many trials, so we'll see. So what about other targets? DNA damage response, epigenetic modifiers. What about other targets, the other new trials that are showing promise in the clinic? So this is Delphi 301, which looks at tarlatamab, which is a DLL-targeted BiTE, right? It's a bispecific T-cell engager. And the phase 2, after 2 lines of therapy, it showed an objective response rate of 40% and the overall survival for 14.3 months. There's a phase 3 trial, which is Delphi 304, that is second-line tarlatamab versus standard of care chemo after first-line platinum-based chemotherapy. This is the trial number. You can check it out. And now there are trials that are trying to bring tarlatamab into the first-line setting. These are the 2 trials that are looking, the Delphi 305 and the Delphi 306, that are looking into tarlatamab versus durvalumab and tarlatamab after chemoradiation in limited-stage small cell. There are trials that are going on in trying to bring tarlatamab in the first-line setting for small cell. Another trial, this was actually presented at LiveLung last year. This is called ifinatamab deruxtecan. God bless the people who name these drugs. But they are B7H3 targeted ADCs in refractory small cell lung cancer, right? It showed an objective response rate of 52.4%. This was a phase 1/2, and it's in the second-line setting, so great response, 52.4%. And then there are these phase 3 IDA2, IDXT standard of care after first-line platinum-based regimen. This is the trial number. You can check it out. You can take pictures and you can check it out, the trials. And then there is a phase 2 now with ID8, IDXT after less than 3 lines of prior lines of treatment, including the first line prior platinum-based chemotherapy. So this is a trial number. You can go and check it out and talk to your physician about it. So this is the clinical trials, and I spoke about targets. What about biomarkers? Are there biomarkers? And I know Christine did a great job of making a comment earlier about non-small cell versus small cell. So non-small cell lung cancer, it is one of our really high-performing cousin. They're doing well in school. They're really getting all the grades. They're going into Ivy Leagues. They have been doing that for a while now. And that's because there are these mutations that we can target. EGFR, KRAS, ALK, ROS, gain-of-function mutations. The patients go into the clinic, they get the NGS testing, they get the mutations, and then there is a drug for them, right? And I'm not going to talk about non-small cell at all because, of course, there are so many challenges there. But at least there is something to go after. In small cell, there are no biomarkers. Mutation-based, at least, there are no biomarkers, right? Not yet, anyway. So this is what small cell really is. It's like, you have a RBNP53 loss. There are these 4 subtypes that the field has proposed. And then there are these markers that these subtypes sort of express, depending upon which paper you're reading. And then there are these targets that you can go after. It's a great news. It's a lot of information there, but it's a great news. We at least now have some information about these tumors, right? And I think small cell is a disease which has really urged us to go back to the lab. It has almost challenged us that go back to the lab, learn the biology, and then design the trials. Because we have to learn the biology first with small cell. It cannot be any other way, actually. And then some of these are hot tumors, some of these are cold, and then within the hot tumors there are subdivisions. I'm not going to go into that, but we are sort of dissecting the biology of small cell bit by bit, right? And this is another target, another biomarker that went straight from the lab to the clinic. So this was a study that was published. Actually, 3 studies came out almost concurrently in 2016, 2017. One from our team at MD Anderson, I was a postdoc there. One from the team at NCI, and the other team was at Memorial Sloan Kettering, right? 3 studies said, SCLC11, it's a DNA RNA helicase. It does really predict response to PARP inhibitor and chemotherapy. That later on, there's a retrospective analysis that was led by Kathy Pietanza that also showed that SCLC11 could be a predictor of response to chemotherapy and PARP inhibitor. Why is this important? Because this study was presented by Nagla Abdel Karim, a very dear friend of mine, was presented at ASCO last year. And she's the lead in this trial, which is called S1929. It's a Walk-sponsored study. And it selected patients based on SCLC11 expression. And they put either atezo or atezo plus talazoparib, right? And when you look at this Kaplan-Meier curve, you're like, not good. We can do better. But what this trial really tells us is there is a feasibility of small cell lung cancer trial that's biomarker-driven. This is the first trial that actually went there and really did a biomarker-driven trial in small cell. I think it's a bold approach, and I think more companies are now getting on board and even attempting to do biomarker-driven trial in small cell lung cancer. Because unless we do a trial, we can keep doing things in the lab, but unless we put it in test, we'll never know whether it works or not. So many, many unanswered questions when it comes to subtyping and biomarkers in small cell. Lineage plasticity, these cancers are promiscuous. What is a biomarker when it changes its subtype? Do we change therapy? Do we not? I don't think we know the answer yet. What is the best way to do it in a setting that works for everyone? If it's only restricted to centers that have zillions of dollars, it won't work, right? It needs to be a biomarker that every center everywhere can adapt, can do, that a pathologist without introducing much resources can actually do it. So a biomarker needs to be dependable, applicable, easily used, right? And it should not drain all the resources of the center. And then what is the best method to do subtyping? Can neuroendocrine subtyping be a surrogate for treatment for small cell lung cancer. And I spoke about Schlafen. So what about, Schlafen low population? We think we do have a target for that. And of course, the only way to do this is prospective studies are required to compare and confirm the validity of small cell subtypes and methodologies in the clinic. There's no other way to do this. I can stand here and make hypotheses all day long, but we need to test this in the patient. And I think I really want to give a shout out to all the pharma partners in the room who are at least sitting with us now to have these conversations about biomarker-driven trial in small cell. I really thank you all for believing in our preclinical research, because I think that will take us forward. So from that black box, we have come to here, and now I'm a part of one, there are others being planned, that where we are doing umbrella trials of small cell lung cancer that are biomarker driven. And I think it's a real way forward where we are selecting patients. Will biomarkers be as simple as non-small cell? No. It will be complex. It will be more than one. But we at least have to try. We owe it to our patients to try. And what I believe I demonstrated, and you can tell me whether I didn't, is there is a cohort of small cell patients that are small but real who have durable benefit with immunotherapy. We need to understand who these patients are, and we need to understand the patients who do not respond. The biology is very important. There is this repression of MHC class 1, which is a major hurdle for these cancers responding to immunotherapy. Someone asked there, why immunotherapy? There was a question from the audience about immunotherapy. And I think part of the problem is this repression of MHC class 1. So all these BiTEs and ADCs that I spoke about, I'm not going to repeat their names, they're too hard, but all these BiTEs and ADCs, they kind of counteract that, right? They bring the cancer cell to your immune cell. So they're counteracting this inherent problem that small cell has, and that's why they're working. We will have to see. There are many things to work out, like the toxicity, applicability, combination, so there are many, many unanswered questions, of course, but at least they're showing preliminary results that are hopeful. And restoration of antigen presentation, I showed you the targets, EZH2, LSD1, so we would have to see. And then, of course, I spoke about DNA damage response vulnerabilities. Personalized biomarker-driven trials, molecular subtypes are important for the biology. Are they important in the clinic? Stay tuned, because when I come back, and I hope I come back, Dusty, in 2 years' time, I'll tell you whether these trials worked or not. And then novel therapies, BiTEs, CAR-Ts, ADCs, we'll have to stay tuned about how they do, long-term response, patients, the toxicities. And I mention toxicities all the time because, we have to remember the journey to get the curve. So that's not trivial at all. So, that's why toxicity matters. And then blood-based biomarkers, I'll leave that to Simon. I'm not going to steal his thunder. He has a lot of thunder. And then biology of treatment-resistant small cell. We are just trying to understand that right now. We're scratching the surface. I think there's so much to unpack there. And I think blood-based biomarkers, single-cell sequencing, all the fancy technologies that I spoke about from your tissues will benefit these trials. Road ahead, oh my God, the first point, improve funding for small cell research. I've seen on Twitter through Dusty going to NCI to advocate for that. I think it's very, very important. I think it's absolutely important. And limited stage small cell, early detection, interception, and identification of individuals at risk. There are many trials coming out, and I think developing and implementing effective treatment programs, liquid biopsy, racial and ethnic disparity in small cell trials. I think identifying and disseminating the knowledge gaps that exist in clinical and preclinical research, we have to identify and we have to implement that. Because it should be a trial that works for everybody. And then incorporating racially and ethnic diverse models in preclinical research. All our models, unfortunately, currently come from a smaller population of patients, right, that don't show the whole demographic of patients. So we have to have diverse populations from where we have collected these preclinical models that can tell us about these disparities. And increasing access and enrollment of racial and ethnic diverse populations in clinical trials. The potential of bispecific antibodies and antibody-drug conjugates, we have to identify whether it would be a biomarker-driven or maybe it would be like chemotherapy. Everyone benefits. And the refinement of toxicity and adverse events when it comes to BiTEs and ADCs. Which line of therapy would they be involved in? And then preventing brain metastasis to improve the quality of life. I cannot say enough about the quality of life for these patients. So how do we study, how do we implement recurring that, going around it? But I want to end on a hopeful note. We have made a lot of progress. It has been untiring work for everyone, like all the researchers and the clinicians that get to speak here, and they're there somewhere remotely. All their work has culminated to this. There are more than 227 small cell trials that are currently open, pending activation in the clinicaltrials.gov. I think this number is already low. 180 are currently recruiting, right? So it's a huge, huge step forward. There are new trials that have a trend of targeted therapies that are not dependent on pathway activation. And I think it will be very important for a cancer like small cell, where mutation is not the answer. Maybe surface targets would be the answer. And then specific targets for small cell, I heard someone about CES6, who's going back and best of luck. CES6, DLL3, B7H3, and others that have really worked. We are entering, and this was something that Tofiko Wanikoko said in small cell lung cancer, we are entering a golden age of small cell lung cancer, where we are really understanding the biology. We are really trying to teach patients based on the biology that we have learned. And we are seeing the differences in the clinic. We'll continue to see that. And none of this would have been possible. This is the work of Lot, so many years, so wouldn't even fit into the slide. But of course, I want to thank the people in my lab, the people at Mount Sinai, all my collaborators. This is a very partial list, my funding agencies, but most of all, you. Because without you, there is no research. But I would really like to say this is a hard journey. This is a hard journey, really. But I think once you choose hope, anything is possible. So I would encourage, and we are trying. I'm like a toddler in a backseat of a car, and I ask my team every day, are we there yet? Are we there yet? Are we there yet? And I will not stop asking that question till we get there. Like, I really won't, because we are not there yet. We are getting there, but I don't think we are there yet, right? So I will keep asking that question, and hopefully trying to get to the answer. But please choose hope. And I think these organizations really, really help you stay hopeful, stay really connected. Talk to us about what we do. If something that we do doesn't make sense, talk to us, because what your journey really orients us, really makes us sort of ground ourselves into thinking what we are doing, how we are doing, and how we can get there faster. But I would really encourage you to choose hope, because I choose hope every day when I do the research. So thank you.
Thank you so much, Dr. Sen. We have probably a few moments for a few questions before lunch at noon. Anybody have okay?
I was just wondering if you are the Dr. Sen that was cited in one of Dr. Oliver's slides on immunotherapy. I saw that there was a Dr. Sen cited on one of Dr. Oliver's slides, and I was just wondering if that was you.
Same Sen.
All right, right on.
Anyone else? Okay.
First of all, thank you for unpacking the whole single cell versus bulk. I mean, it's really exciting to think about the opportunities with single cell versus this slurry of data. But also, thank you for the imagery. I love Jack Sparrow taking on, the whole T-cell thing. That was very cool. But here's the most important question of the day. In your logo, the Sen Lab, what was the inspiration there in your logo of the Sen Lab?
What was the inspiration? So there's New York, there's DNA, there's the lab things. Well,
I told you it was a hard one.
I know, you're asking me all the hard questions. I don't know, I mean, I really don't know. I think I brought that all together to tie into a loop. I think it's an infinity loop because I feel it's truly an infinity loop. We go back and forth in this dialogue from the lab to the clinic to the lab, and we're trying to sort of bridge the gap and close that loop together so that we can always learn from each other, because I don't think we will ever progress without learning from each other. So I think that was the inspiration. But as I said, Jim, you know me, I'm a small cell nut. So, it's really a privilege to be doing what I do. Yeah.